Order! Order! You are an incorrigible delinquent at times. Behave yourself, man!
Optogenetic treatment of neurons spawning dendritic spines to form synaptic connections. This is neither a Scrabble crib sheet nor a sesquipedalian fantasy sequence, but rather a process by which memories previously believed to have been banished by Alzheimer's disease can now be retrieved. New research at the Pikawa Institute of Memory and Learning at the Massachusetts Institute of Technology has been conducted on mice with Alzheimer's-like symptoms. But the ramifications for humanity could prove considerable. Dr. Susumu Tonegawa is the research leader and a former winner of the Nobel Prize for Medicine. He joins us now from MIT. Professor, what have you been doing and what are you most excited about discovering?
Okay, in the development of Alzheimer's disease, it's been known that before the typical signature, cellular signature of this disease appears, like plaques and tangles, there is a period of 1 to a few years where patients already show memory impairment. Many researchers and doctors have thought this is because these patients cannot form a new memory. But we showed in our mouse model of early Alzheimer's that actually these mice can form a memory perfectly well. It's just the problem is they cannot retrieve the memory. So that's the most exciting finding. And the second exciting finding is we could cure this impairment of memory recall by increasing the connection of spines that is on the memory holding cells. And this way, with just a natural recall cues, the mouse showed normal retrieval of memory.
I am almost certainly going to embarrass myself now, Professor, but you create these spines by shining a special type of light onto them.
Yes, that's exactly what we did. So this is a technology called optogenetics. So these memory holding cells are labeled with a light-sensitive protein called channel rhodopsin. And by applying appropriate strengths of light, blue light, that we could increase the spine density of these memory-bearing cells.
The extension of this technology to human beings is within reach or not?
Well, that depends on the technological development. And among the experts, the opinions are not the same. Some optimistic people will say some kind of therapy based on this type of principle can be done within the next few years, maybe 3 or 4 years. But some more conservative engineers and brain researchers would say they would take longer than that.
What would you say, Professor?
Oh, I'm not really expert on this because I'm a basic brain researcher. I'm not an engineering person. But I would be cautious. Eventually, this type of research will provide us the precise information about where in the brain the brain cells should be activated. So I would be conservative, but eventually, I think this type of experimental result will help for engineers and surgeons to target the specific small area of the brain to stimulate in order to reduce the disorders.
Okay. So, Dr. Susumu Tonegawa, many thanks indeed, and congratulations.
Thank you very much.
Thank you.